The overall goal of this procedure is to investigate intracellular mechanisms that control bacterial dissemination during infection in vitro and in vivo by using tissue culture cells and zebrafish larvae. The in vitro method begins with preparation of Shigella flexneri, a gram-negative entero-invasive bacterial pathogen. HeLa are then infected and siRNA or pharmacological reagents are used to manipulate the host autophagy machinery and cytoskeleton during infection. To study autophagy-cytoskeleton interactions caused by Shigella in vivo, zebrafish embryos are infected 3 days post-fertilization. For either method, the final step is to label tissues with autophagy and cytoskeletal markers. Ultimately, results will provide insights into the mechanisms required for the control of infection by cytosolic host responses.
The advantage of using Shigella to study bacterial autophagy over other bacteria, such as Listeria, is that Shigella-targeted autophagy can also be entrapped in septin cages.
This method can help answer key questions in the autophagy field, such as, what is the role of the cytoskeleton in autophagy? And what is the role of autophagy in the restriction or promotion of intracellular bacteria?
The implications of this technique can extend towards other processes of autophagy, such as the autophagy of viruses or mitochondria, and can also have therapeutic implications for human diseases with a septin or autophagy component.
Visual demonstration of zebrafish infection by Shigella is critical, as the steps involving injection of zebrafish with bacteria are difficult to learn, because needle preparation and proper injection takes time, and high resolution microscopy of zebrafish infection takes experience.